Hello students, welcome to my biology class. Today's class is for students of class 12. I will be discussing the fifth chapter, that is Principles of Inheritance and Variation. Under this chapter, we already have discussed certain things or different areas and topics. Today's topic is linkage and crossing over. What are we going to learn here? Or at the end of this discussion, what you will be able to learn? These are the learning objectives. So, you'll be able to learn and define certain terms like linkage, crossing over, even independent assortment. You'll be able to write what are the conditions for linkage and recombination to occur. You'll be able to relate independent assortment, linkage, and crossing over. You'll be able to apply your skill to solve problems on crosses involving sex chromosomes, because earlier to this, all crosses were on autosomes. You'll be able to explain the frequency of recombinants and how this frequency reflects the strength of the linkage. You will explain how Morgan proved that linkage and crossing over occurs between genes. Now I'm going to show you a photograph. See this photograph carefully. Can you identify what is this? Yeah, I suppose it's not a very difficult question. But why did I show this question? I will ask this question again at the end of this discussion. And I hope by this time you will realize why I have shown this photograph. One important scientist related to this discussion is Thomas Hunt Morgan. Thomas Hunt Morgan, will know about him and his work. You can see here, it is the Drosophila. And this is a laboratory where Drosophila has been cultured. And also on the starting page, you must have seen a color photograph of a Drosophila. Drosophila, or the fruit fly, is a item, is an experimental material, is a model experimental material, and is linked to the name T.H. Morgan. Now, let us know about T.H. Morgan. Thomas Hunt Morgan was born on September 25, 1866 in Kentucky, USA. He began his work on fruit fly in 1909, and he created a fly lab in Columbia University. And this is very, very well known and very famous laboratory in Columbia University. And because of his experiments and findings, Drosophila melanogaster, this fruit fly, will always be known, will always be associated with the name of T.H. Morgan. Morgan's first papers dealt with demonstration of sex linkage of the genes of white eyes in the fly. Now, this is very important. In this fly, that is Drosophila, the red eye is normal and the white eye is a mutant, which Morgan first observed. His work also demonstrated that the important fact that spontaneous mutation frequently appeared in the culture of the flies. So, among the red-eyed Drosophila, he frequently found some white-eyed, and that was the main interest which developed, and that was the basis of his work. Morgan, he put forward a theory of linear arrangement of genes in the chromosome. That means genes are in a straight line, means they are arranged in the chromosome. Expanding this theory in his book, Mechanism of Mendelian Heredity in 1915. Morgan wrote many books. Heredity and Sex in 1913, Physical Basis of Heredity in 1919, Embryology and Genetics, and many other books. Experimental Embryology, Scientific Basis of Evolution, and many others. T.H. Morgan was awarded the Nobel Prize in 1946 for his production of mutations by the means of X-rays. And finally, he died in the year 1945. Today's discussion totally is based on the work of Thomas Hunt Morgan. Now, why Drosophila was chosen as a model organism? And this is a very, very unique and very good experimental material. Why it is so? Compared to the pea plants, where only you can have two generations in a year, here within weeks you can have generations. It is easy to culture in synthetic medium, and it takes only 2 weeks to get the results. Easy to get it, and a large number of progenies are produced as a result of single mating. Easily distinguishable, you can see here also, the female and the males, they are easily distinguishable. So, easily distinguishable male and female flies. The characters under study are very, very clear and visible. They have only 8 chromosomes in the diploid cell, and all the chromosomes are distinctly different. Now let's see what was actually Morgan's work. You please listen carefully. I will try to explain as many times required. Morgan carried out several dihybrid crosses in Drosophila to study that genes were sex-linked. Here we are coming to a new term, that is sex-linked, which means linked to the X chromosome. Mendel's finding differed from Morgan's finding. The basic reason is Mendel's work was on autosomes, Morgan's work was on sex chromosome. So you will find in the latest half also you will find that in Morgan's experiments, the reciprocal crosses did not give the same results. Why? Because the sex chromosomes are different in males and females. In case of Mendel's work, there was no such problem because the genes were, or the factors, Mendelian factors, they were located on the autosomes. But here, since males are heterogametic and females are homogametic, their expression differs, which we'll discuss in the later part. The crosses were similar to the dihybrid cross carried out by Mendel in peas. Now, what Morgan did. Morgan hybridized yellow-bodied, white-eyed females. Now here you see, nowhere in Mendelian experiments you have seen describing as this male and that female, because reciprocal crosses were giving the same results. So here, expression of the gene in the male and females, they differ. That was the basis to understand that this is not an autosomal inheritance. This is a sex chromosomal inheritance. So here, Morgan, he hybridized yellow-bodied, white-eyed female to a brown-bodied, red-eyed males and intercrossed their F1 progenies. He observed that the two genes did not segregate independently, and the F2 ratio deviated from the 9:3:3:1 Mendelian dihybrid phenotypic ratio. Again, let me remind you, why this? Very simple. There are various factors. In Mendelian experiments, the two genes were located on two different chromosomes. The two genes were located on autosomes, which is not the same in Morgan's work. What was the observation? Morgan and his group knew that genes were located on the X chromosome. So, he was aware that when the two genes in the dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental types. Very simple. When the gene A and B are located on the X chromosome or in a particular chromosome, then the chance of independent assortment is very negligible. In case of Mendel, A and B were on two separate chromosomes. But here, since they knew that it was on the same chromosome, so non-parental type, non-parental combination, the chances were very, very low. So Morgan attributed this event to be physical association. That means the two genes have a physical association. In Mendelism, there was no such physical association because they were on two separate chromosomes. But here, there is a physical association, or he later said as linkage. Linkage of the two genes, and so he coined the term linkage. Because the parental frequency was much, much higher than the non-parental types, the reason was the physical association between the two genes. That means the two genes were on the same chromosome. And to describe this physical association of genes on a chromosome and the term recombinant. Now, so two terms now come into existence. One is the linkage, another is the recombinants. Who are those recombinants? New combinations formed, which are non-parental gene combinations. I think it is now clear. Now, recombinants also was obtained in Mendel's dihybrid ratio. If you just think of 9:3:3:1 phenotypic ratio of the F2, then 9 and 1. These two were parental combinations. In between 3 and 3, they were, of course, these two were recombinants. But here we find a large variation in the frequencies. Morgan and his group found that when genes were grouped on the same chromosomes, now they have come to the conclusion that there is some linkage, there is some physical association. Now again more observations. What is that observation? Even when the genes are located on the same chromosome, some genes were tightly linked, whereas some genes were not so tightly linked. Now, when there was tight linkage, then recombination frequency was low. But if the genes were loosely linked, then the frequency of recombination was high. These are certain points which you have to remember. Otherwise, at the end of the discussion, you will not be following what actually happened or what actually we have discussed. That is, when we consider that a linked gene or chromosomes on the same chromosome, genes on the same chromosomes are called as linked genes. I think it's clear. Since on a single chromosome there are many genes, so genes which are adjacent, very close, they are more tightly linked. A gene on the far ends, two ends of the chromosome, they are at a much greater distance, which shows that when the genes are loosely linked occurs when there is a greater distance between the two genes which are under consideration. So when there is a more distance between the two genes, then that means there is loose linking. And under such circumstances, they show higher frequency of recombination. I think it is now clear. Genes which are close, they are tightly linked, they will show low recombination frequencies. Genes which are at far off distances on the same chromosome, they are loosely linked and show higher frequency of recombination. Now, what Morgan found is that the genes which were tightly linked, they showed only 1.3% recombination. Whereas in the other set of experiment where they were loosely linked, they showed 37.2% recombination. We'll see the experiments and the crosses also. Let us see the cross A. Try to understand this. He crossed female with yellow body, white eye. So female is yellow body and white eye with a wild type male, that is grey body and red eye. Now here let me tell you the sign conventions. These are mutants. In sex-linked inheritance, we give more importance to the mutants. So here the mutant is yellow body, white eye. So we have taken the letters Y and W, and corresponding their normal traits as with a positive sign. So the wild type here means normal. The normal is grey body. It is Y plus. Red eye is W plus. In Mendelism, we did the different thing. The dominant genes, we took the letters for the dominant genes. Here we do the reverse work. We add a plus sign to identify the gene as a wild gene or a normal gene. Now, when he made such a cross, what he found? He found that all the female individuals were wild type, but the males expressed mutant traits. Interesting. Here we are not talking of the whole population as a whole. We are now segregating the population into males and females and then comparing their frequencies. Because reciprocal crosses do not give the same results in such crosses. When F1 males and females were crossed, now see, F1 males and females, this is another dihybrid cross, two characters are being studied. What we find? 98.7% were parental type and very, very low, that is 1.3% were recombinants. Now, how we got such a result? Let us try to understand this. See the cross. Here you have to understand X chromosome, X chromosome. Since this inheritance is sex linked, so we consider XY chromosomes. Females are XX type, males are X and Y type. Here the colors are taken different. Reason being to make it clear of their movement, to realize which chromosome goes where. So female, since this is a recessive trait, so it is yellow-white, so this has to be in homozygous condition. White also is in homozygous condition. In case of males, no gene on the Y chromosome. So single dose of a gene expresses the trait in the males here. So a single copy of Y plus will give the wild type expression. Single copy of W plus will give the wild type of expression. Now what happens? If we make a cross between these two, what we get? As per Mendelism, this X chromosome, they separate. This X chromosome and this Y chromosome separates. Since this is a homozygous condition, so from this only one type of gamete is produced, that is this yellow colored X chromosome with Y and W. So one type of only gamete is produced. Here two types of gametes produced. Now don't think that here there is only W plus and W plus and Y plus, so one type of gametes can be produced. No, because X and Y are different, so they separate. One small y, small w pairs with Y plus W plus. Here you can see. So one of the chromosome from the female comes, one X chromosome from the male comes, and this is a female with a wild type. Why? because Y plus, W plus, they are dominant over these recessive mutants. What happens to the males? The Y chromosome and one of the X chromosome pairs. X chromosomes bears the mutant genes. So naturally, out of this population, all the females are heterozygous, but they are all wild type. All the males here are yellow and white type, means mutants. Because I said earlier that a single dose of the gene in the males will express. Now let's see what happens. F2 generation. Now let's study the F2 generation. Now if these are crossed again, these two are crossed. Then this one separates, this chromosome separates. This is independent assort. This is segregation. This separates, and similarly this also separates. So now what is the possibility? There can be 4 different combinations. This chromosome from the female can pair with this chromosome. Let us study here. We are now going to study all the probabilities. It is a question of probabilities. Now let us study what happens. We'll study this. Now these 2 chromosomes, they will be separating. Clear? These 2 chromosomes, they will be separating. Now during this separation, what you will find? This chromosome will pair with this chromosome. This chromosome will pair with this chromosome. Similarly, this chromosome will pair with this chromosome, and this chromosome will pair with the Y chromosome, like this. So we have 4 combinations. These are the 4 combinations. Let us see this one. How this is formed? Y plus W of the female pairs with the Y chromosome. This one is done. Now this Y chromosome has already paired with this one probability. This Y chromosome can pair with this one also. W and Y, you got this one. So this is over. Now the X chromosome of the male. This X chromosome of the male can pair with this one, Y plus W plus. So here you get it, Y plus W plus. This X chromosome of the male can pair with this X chromosome of the female. So you get this one. Okay? So these are the possibilities. There are more possibilities. What are those possibilities? Now suppose crossing over occurs. If crossing over occurs, then what will happen? That is, crossing over here will occur in the XX chromosome. It will not occur here. Here it will occur. That is between Y and X. You see, Y and X are very close to each other. Okay, Y and W, sorry, not X. Y and W are very close to each other. Still, suppose there is crossing over in between. Crossing over in between. Now recall the concept of crossing over in meiosis 1. That is reciprocal exchange of chromosomal segments between non-sister chromatids of a homologous pair of chromosomes. Now these are non-sister. These are non-sister chromatids. This is yellow and this is blue. So they are non-sisters. Now between these two, so accordingly, in meiosis 1, here there will be one more chromatid, here there will be one more chromatid. Forget about those. Now we will consider only this much. So here there is crossing over. Crossing over follows by chiasmata formation, breakage and reunion of the chromosomal segments. So here there is a breakage and reunion. So this part, blue part, goes and attaches here, and the yellow part comes and attaches here. You see here it has happened. The blue part, that is Y plus, comes and attaches onto this chromosome. So it becomes Y plus W. And another chromosome. What happens to this one? This one becomes, because this yellow part, that is Y gene, comes and attaches to the blue part. So here you see. So now these two are crossed over. This is the result of the crossing over. So it has yellow part and a blue part, and these have major blue part and a small yellow part. So now you see there is a difference in the combinations. Here it was Y and W, it was W plus and W plus. Now it has become Y plus W and Y W plus. Now if this is the condition, such a crossover female will be allowed to cross with this male. Then what will happen? Just as we said, this Y chromosome pairs with this one, Y chromosome pairs with this X chromosome. Similarly, this X chromosome of the male also pairs with this X chromosome of the female. Like this we have discussed here. Now this thing will happen with this one. Let us see what happens. Y plus W, this X chromosome pairs with the Y chromosome. Here you see, this is the chromosome pairs with the Y chromosome. I think it's clear. Similarly, Y and W plus, this blue and yellow, pairs with Y chromosome. You can see here. Similarly, the X chromosome of the male pairs with this one. See it here. This X chromosome of the male here, and this X chromosome of the female is here. And again, this X chromosome of the male and this X chromosome of the female comes here. So this is the result, probable results when there is a recombinant. When there is a recombinant. So what Morgan found is that in his population, these are the possibilities. These are the possibilities. Among his possibilities, he found that the parental combinations were very, very high, 98.7%. Recombinants were 1.3%, which means, I told you that you see here, Y and W are very close. That is why recombinants are low. Because chances of crossing over between these two is low. Had this been in this location, Y and W at far off location, then the chance of recombination would have been higher. We'll see in the next experiment also. I think this is clear very much. Okay, thank you. Let's move on. So, I have understood how the recombinants occur and how the parental combinations occur. Let's move on. In cross B, Morgan crossed white-eyed females with miniature wings. Now he have taken two different characteristics. In the previous one, the characteristics were different. Now we take a different characteristic. That is white-eyed. I told you that white eyes are always mutants. These are recessive mutants. And miniature wings. This is also a recessive mutant. They were crossed with wild type. Females were having the mutant genes, and the males having normal genes or the wild genes. What he found? In the F1, he found all females to be wild type. Now, I think you can understand why the females are all wild type here. Because the male itself is wild, and this wild male means normal genes in the X chromosome. This normal gene of the X chromosome pairs with the recessive mutant of the females. Which one expresses? The wild type expresses. That is why the females are normal. But in case of males, male becomes mutant. Why? Because male have only one chromosome, that is X. Y chromosome, no genes. So whatever is there on the X chromosome will express in the males. So a single dose of the gene expresses in the males, whereas in the females you require 2 doses of the mutant genes to express. So in males, mutant genes expresses more easily. In this cross, he found that 62.8% retained parental combination, only 37.2, not only, but here on higher levels. It is 37.2 were recombinants. much higher than the previous experiment which you have just studied. Why this? Already I have discussed that this is because of crossing over and chiasma formation. We'll again try to explain you this experiment. As we have explained in the cross A, we'll try to explain you the cross B. Let us see here. This is cross B. Now recall this. In the previous case, the two genes were very close. Now the two genes are quite apart. Two genes are quite apart. W, white, miniature, M. So these are mutants. So the letters chosen will be white W, miniature M. And their normal wild will be W plus and M plus. This is the sign convention for sex-linked inheritance. Please remember. Here we don't use capital W, capital M, small m as we are using in Mendelism. That is for autosomal inheritance. Now, what happens? Here, this female, these two X chromosome segregates. This two during gamete formation they will separate. These two will separate. And since these are homozygous condition, so only one type of gamete is formed. This is heterozygous condition. It is not heterozygous. This is X and Y, two different chromosomes. So it will form two possibilities. One X chromosome of the male pairs with the X chromosome of the female, and this is the result: W plus W, M plus M. So what expresses? Naturally W plus and M plus, because W and M, these are recessive mutants. Their wild types are present. So the expression is the wild type. Whereas in this case, the Y chromosome of the male gets a X chromosome from the female. The X chromosome bears W and M. This is the mutant genes. So this male will express white and miniature. So white-eyed and miniature wings. I hope up to this much there is no problem in understanding. If you have any problem in understanding, please rewind, go back and try to understand again. Now what happens? This is the F1 result. Now let's see what happens in the F2. The same thing, nothing different. You'll be able to understand. This is the same thing which we have discussed in the previous case. So F2, these two separates, these two separates. So from here two types of gametes, from here two types of gametes, and so ultimately you have four combinations. This Y chromosome of the male can pair with this yellow chromosome. Here you can find the Y chromosome pairs with the WM female X chromosome. This Y chromosome pairs with the W plus M plus X chromosome from the female. So these two types. Now the X chromosome from the male pairs with this blue one, as you see here, and again this yellow chromosome pairs with this yellow chromosome. Both are X chromosome. It is here. So what we find? The males, 50% of the males, because there are two males and two females. Always remember that in sex-linked inheritance, always the result will be two males and two females. So when you are going to write the ratios obtained, you must write separately what happened in the males and what happened in the females. Don't write in total that out of 4, 75% is this and 25% is that, or 50% is this. Don't write like that. You may write, but even after that, you must mention that among the males, the ratio here is among the males, because always in the population 50% will be male and 50% will be female probability. So among the males, 50% is wild type, you see here, because one wild and one miniature. So 50% is wild type and 50% is white miniature or mutant type. I think it's clear. Similarly, among the females, what do we find here? Again, 50% is white miniature, is the mutant, and 50% is wild type. If you want to add both this, what do you find? Out of 4, 2 are wild, 2 are mutants. But this is not the way to express the sex-linked inheritance. Next, this is one probability. Another probability which is very important. Now I am discussing this because how Morgan got such results. Now here in his results he found that comparative to the previous experiment, the parental combinations were less, and comparative to the previous experiment, the frequency of recombinance was much higher. How this frequency of recombination was higher in this case, to explain that we shall continue. That is, suppose crossing over takes place, then what will happen? Crossing over occur between these two, as we have seen in the previous case. I think it's clear. I don't need to explain again. So crossing over occurs in between these two, and there is exchange of chromosomal segment. During this chromosomal segment, these genes are interchanged. So the new combination is, here it was W and M, it becomes W plus, this part goes here and this part goes here. So half yellow, half blue, W plus M. And the other recombinant becomes W and M plus. So half yellow from this one comes here and forms W and M plus. So this combination we obtain, probability. Now this female will cross with this male. These two separates. These two separates. So again, if you make a Punnett square, you plot them and you get 4 results. Okay. So this Y chromosome pairs with this one. This Y chromosome pairs with this one. In the other part, this X chromosome pairs with this one, and this X chromosome pairs with this one. Then what will be the result? Let us see now. This is the result. What is this? See, Y chromosome pairs with this one. Is this one. Y chromosome pairs with this one. WM plus. Y chromosome pairs with W and M plus. This is the result. Next is this X chromosome, yellow one, pairs with this one. Okay, this one, yellow one, pairs with W plus M. So this is the result. Similarly, WM, this yellow chromosome, pairs with WM plus. WM plus, and this is the X chromosome from the male. This. So what we find here, miniature. Out of the 2 males, one is miniature, another is white. Okay, here what Sean expresses? Is here whatever is there is going to express white, and of course M plus is wild. So here the mutants, I am talking of the mutants expressing. Okay, because W plus is normal. Our interest is with the not the normal. Our interest is with the mutants. So here M is the mutant, W plus is the normal. So the mutant here expresses. Here W is mutant, M plus is normal. So here we are interested with the expression of the mutant, that is white. Similarly here. So this is the observation when there is a crossover between W and M. Now what he found in the population taken together, these are all probabilities. What he found in the population studies, he found that here the parental combinations reduced. 62.8% were parentals, whereas 37.2% were mutants. Sorry, it should be called as recombinants. Why there was an increase in the percentage of these recombinants is a matter of question. Again, I tell you, it is the distance. So as the distance between the genes increases, the probability of crossing over also increases. When the probability of crossing over increases, the recombinant frequency also increases. This is what Morgan tried to explain. I think it is now clear from this process. So now before we end this discussion, again let me remind you of certain things. That is, three terms are very important. One is independent assortment. In Mendelism, we have studied that when genes are located on the separate chromosomes, then they assort independent of each other. And when they assort independent of each other, then certain recombinants do occur. In dihybrid test cross, it can be clearly visible the test cross in a dihybrid is 1 is to 1 is to 1 is to 1. That is equal proportion of parental and recombinations. Recombinants 50%, parents 50%. Deviation from this will be explaining in a different way. If there is linkage, then the parental combination 50% will be getting higher. If there is recombination, if there is linkage, then never the recombination will be 50%. It will go lower. Depending upon how much linked the genes are, the tendency will go towards zero. So more deviation from 50% recombination will show an increasing strength of linkage. So when there is more distance, there is less linkage. There is more chance for crossing over. This is very important. So based on the frequency, crossover frequency or the recombination frequency, we can justify the distance between the two genes. I think now you've understood this part. So now again we come to the first slide which we have shown you, that what do you understand from here. Now let me tell you, these are all trains. Consider each, what is this? These are all different coaches. Different coaches as a gene. The whole train is a chromosome and every coach is a gene. So wherever the engine takes the train goes together. The first coach and the last coach reaches the same destination because they are all linked. All the coaches are linked, like all the genes are linked on the chromosome. So the whole train is a chromosome. Wherever the train goes, all the coaches goes. Because here there is complete linkage. While the train is moving, for example, in between, in between this linkage, this link opens, then what happens? Suppose it opens here, then this part will not go to the destination, above part goes to the destination. Then this part has to be joined with some other train to go to certain destination. Crossing over occurs. Now, if I ask you a question, here you are sitting in this coach. Your friend is sitting in this coach. So the chance that you two be separated during the journey will be much less than your another friend. You are sitting here, your friend one is sitting here, and your another friend is sitting here. Just compare. So the chance of you two sitting side by side in coaches, your chance is less than your chance of separation from this friend. Why? Because in between there are several points where the breakage can occur. And here it is only one. So suppose there are 10 such breakage, your chance of separating your first friend is only 1 out of 10. Your chance of breaking between these two is much more because there are more points in between where breakage can occur. So the chances is more. That is why I have put this figure. This is my photography, and to show this, that the train is a type of linkage, and wherever the engine goes, the whole train goes because the coaches are linked. At various places, at various places, some of the coaches may be detached and attached to another train to take to different places. That is crossover. To make this more simple, I have taken such an example. Hope you have understood crossing over. You hope you understood linkage and also recombination. Now it's time that you take some questions home. So these are some of the questions to define the term linkage and crossing over. Write the conditions for linkage and recombination. How the frequency of recombination reflects the strength of linkage, as I have explained you. What is the relation between recombination and crossing over? Understood? Actually, recombination is crossing over. So recombinant and crossing over, or you can say between linkage and crossing over. You can make any questions. How Morgan proved that linkage and crossing over occurs between the genes. There are many more questions. You can visit my website also to get more questions. And for the moment, you do these questions, you write the answers. And I'm very thankful to you that you have listened to me, and we will be discussing some more topics on chromosomal aberrations. These chromosomal aberrations can also occur on the strain due to various numbers. We'll discuss in the next discussion. Chromosomal aberrations are structural and numeral. We can change the number of coaches. We can change the composition of the coaches to explain chromosomal aberration, or also called as chromosomal mutations. So thank you very much for listening to this session. I hope it has been beneficial to you. So do continue listening to my classes. Thank you.